Jai Hind, dear children. Welcome to today's biology class. I hope all of you have studied the different methods of asexual reproduction that we have discussed last class. Today, in this class, we can learn about another method of asexual reproduction, that is vegetative propagation in plants. It is a method of asexual reproduction in which new plants are obtained from the vegetative parts of the plants. Roots, stems and leaves are the vegetative parts of a plant. The flowers are the reproductive part of the plant. So, in this method, the parts other than the seeds are used as propagules. What are propagules? Propagules means the buds or the structural unit of reproduction other than or instead of seeds. This method of propagation occurs in plants which do not have the capacity to produce seeds or which produce non-viable or non-fertile seeds. Vegetative propagation in plants occurs naturally and can also be done artificially. Here, first we can discuss about the natural methods of vegetative propagation. For example, banana, rose, pineapple, jasmine, etc. In case of banana, if we observe the fruits, we can see small black colored structures as seeds, but they are not fertile seeds. New plants cannot be produced from these seeds. In rose plants also, flowers are produced, but they do not produce seeds. In jasmine plants also, flowers are produced, but they do not produce seeds. Pineapple also do not produce seeds. These plants cannot reproduce through seeds. So, in these type of plants, the new plants are produced from the vegetative parts like stem, leaf and roots. And this process is called as vegetative propagation in plants. The most common natural methods of vegetative propagation in plants are the tuberous roots of some plants develop into new plants. Buds on the roots of some plants can grow into leafy shoots, and the underground stems or modified underground stems like rhizomes, tubers, comb and bulb develop into new plants, and the adventitious buds on leaves can develop into new plants. Tuberous roots are the thick fleshy storage roots. These are true roots which are so thickened and resembles like a tuber but without buds and leaves. These tuberous roots of certain plants give rise to new plants from their fleshy roots. For example, sweet potato, dahlia, etc. Adventitious buds on roots can grow into leafy shoots. In some plants, additional buds are formed on the roots, and these buds can develop into new plants. Some plants like guava, neem, etc. produce flowers and seeds. Then also they produce adventitious buds in the roots, and these buds develop into new plants. Adventitious buds produced on the leaves of some plants may develop into new plants. In some plants like bryophyllum, begonia, etc., buds are produced on the notches along the leaf margin, and these buds, when fall on the ground, develop into new plants. In some plants, the stems are seen underground and they are modified into different forms like tubers, bulb, rhizomes and combs. In some plants, underground stems are modified into enlarged structures in which nutrients are stored. These structures are tubers. Potato is the best example of a tuber. The notches present in the potato are called eyes or buds, and these buds present on the tuber develop into a new plant. Bulbs are modified shoot that has very short stem and an apical and auxiliary buds. These buds grow to form new shoots. For example, onion, garlic, etc. Rhizomes. Rhizomes are thick and fleshy stems which has distinct nodes, internodes, and develops auxiliary buds. New branches develop from these buds which later separate to form new plants. Ginger, turmeric, lotus, etc. are the examples of rhizomes. Corms. Corm is a condensed form of rhizome and consists of a solid and fleshy underground stem bearing one or more buds. Some of these buds produce daughter corms which later grow into new plant. Banana, colocasia are examples of corm. So these are the natural methods of vegetative propagation. Next, we can discuss about the artificial methods of vegetative propagation. The process of growing many plants from a single parent plant by man-made methods are called as artificial propagation. Some of the common methods of artificial propagation in plants are cutting, layering, and grafting. First, we can discuss about cutting. In cutting, a small part of the plant is removed by making a cut. This cutting having some buds is taken and the lower part is buried in moist soil. After a few days, it develops roots and grows into a new plant. Rose, bougainvillea, grapes, sugarcane, cactus, etc. are some of the plants which propagate by cutting. The next method is layering. Layering is done in shrubs and medium-sized plants. In this, one or more branches are bent close to the ground and placed in the soil, and the growing tip remains above the soil. After some days, the roots are developed and the branch develop into a new plant. Layering are of two types: mount layering and air mount layering. Mount layering is done in small shrubs like jasmine, strawberry, gooseberry, etc. Air mount layering is done in plants where stem cannot be bent and a branch is scraped and wrapped with damp moss or mud, and the polythene sheet is used to cover to keep the mud moist. After some days, the roots are developed and the branch developed into a new plant. The next method of artificial propagation is grafting. Grafting is the technique used to join parts from two or more plants so that they can appear to grow as a single plant. In this method, a branch of a good quality plant called as scion is placed over the root system of another plant called as the stalk. Both are tied up such that the chemical activity of the scion and stalk allows them to join together. Sometimes the different varieties are grafted on the same stalk. For example, on citrus rootstock, varieties like sweet orange, lemon, etc. are grafted. Another method of grafting is bud grafting. Bud grafting is done in certain plants like pears, peaches, plum, rose, etc. Another method of vegetative propagation in plants is tissue culture. It is the method of developing plants from a mass of tissue. It is the modern technique of vegetative propagation. Here in this, a piece of tissue is cut from a plant and kept in suitable medium of nutrients. This method is used for obtaining virus-free plants in banana, potato, sugarcane, etc.
Tissue culture. In plant breeding, the technique of tissue culture has emerged as a very efficient tool to propagate better improved crops. Tissue culture is much approachable technique than other traditional plant breeding techniques. By using this method, Cells, tissues, etc. are maintained and grown in vitro under aseptic conditions in artificial medium, providing necessary nutrients for growth. 1950s, scientists found that any part of a plant grown in nutrient media under sterile conditions can give rise to a whole new plant. Technique of tissue culture. The first step of tissue culture is excising or cutting a small part of plant tissue, like the leaf, root, or shoot of a plant. This cut-out part, explant, is then placed in a suitable nutrient medium containing a carbon source such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. The explants are allowed to multiply and form a group of tissues called callus. Callus is irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Tissue culture is performed using a small part of a plant to develop a large number of plants in a short duration. It is thus also called as micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called soma clones. Micropropagation is extensively used in horticulture and floriculture and is employed in the large-scale production of tomato, banana, apple, etc. This technique can also be used to propagate high-yielding disease-resistant varieties in a short span. There are many advantages of tissue culture, which are as follows. A large number of plants can be grown in a short period of time. Healthy plants can be grown from diseased plants. Plants without seeds can be multiplied. It is useful where sexual reproduction is not possible.
This method of vegetative propagation has some advantages as well as disadvantages also. All the plants produced by vegetative propagation are genetically similar to the parent plant to have all its characteristics. By vegetative propagation, we can produce many new plants from just one plant quickly without waiting for flowers and seeds. The plants which do not produce viable seeds are propagated by vegetative propagation. The plants raised by vegetative propagation bear fruits and flowers earlier than those produced from seeds. It does not produce new varieties so that the new plants may find difficult to survive in changed environment. If a parent plant is of poor quality, the offspring will also be a poor quality. It leads to overcrowding around the parent plant so that the plants compete for nutrients and space. What are the advantages of asexual reproduction? In asexual reproduction, only a single parent is needed. It takes less time than the sexual reproduction and more number of offsprings can be produced in shorter time. The offsprings are the exact clones of their parents. The desirable characteristics can be easily incorporated into plants with artificial vegetative propagation. These are some of the advantages of asexual reproduction. I hope you all have understood the contents that we have discussed today. So, read the textbook thoroughly and prepare the notes. Thank you.